Dr. Michael, I couldn't agree more. I think we, as far as confusing the general public, that horse left the barn so long ago. And I don't even think the messaging about vaccines are really good at stopping you from getting really sick and dying. They're just not as good at stopping infection. But, would you rather have COVID or would you rather be dead?
Yeah, trust me, I lost my only sibling to this disease, and it
I'm so
was
sorry.
about 3 weeks before the vaccines became generally available, and about 3 weeks before the first monoclonals became available. And it's just, so I get it. People now are saying, well, these vaccines don't work, and I'm like, it would save my brother, So that's and that's how vaccines traditionally have worked. They've worked not by blocking infection, they work by down modulating disease. And there's just a lot of people don't understand. And I agree with you. We have done a poor job at translating that, but it's also, we're doing it in the context of a historic pandemic at a time of, the deepest divisions I've seen in the United States since the Vietnam War era. It's just a lot of other things in play, right? So difficult.
Yeah, it's really, really difficult, but it's very interesting to hear your perspective, and I'm super excited to hear about your work. I'm just checking. I think we need a little more time, and I apologize about this. Give people maybe 2 more minutes, and even if we don't have a lot of participants, we'll make sure that this recording is transcribed into notes and circulated amongst the members of the CAAB, or we'll show them the recording.
Yes. And we are requesting, we are expecting around 30 people that are registered. But we know that sometimes as vaccines on paper, the efficacy is a number, and then engagement sometimes is another, because that's life.
I'd say 1 more moment, and then I'm going to introduce you and invite you to take it away.
Yes, ma'am.
And I don't know if you can see the chat, but one of our very keen participants oh, you did see it. I see you responded. Great.
Yeah, Kate Hankins is one of my moral compasses in the world.
You're a lucky man.
Yes, I am.
I'd like her to be one of mine too. All right, I'm going to go ahead and introduce Dr. Nelson Michael. My name is Tracy Swan. I am an activist and global health consultant working with ITPC and others. And today we are very lucky to have Dr. Nelson Michael, who's an infectious disease researcher. He's served in the US Army for nearly 30 years, beginning at the Walter Reed Army Institute of Research's Department of Vaccine Research, Division of Retrovirology. Dr. Michael actually directed the US military HIV research program from 2006 until 2018, and has also been directly involved with significant advancements in understanding the pathology of and vaccine development for diseases like HIV, Zika, Ebola, and more, which brings us to COVID. We're all really excited to hear about this vaccine that you're working on. So please take it away.
All right, thanks so much, Tracy, and it's great to be with you again. As you've heard, I've grown up in a couple of tribes. One is the HIV vaccine tribe, which I'm still heavily involved in, and even though I moved up out of MHRP, the Military HIV Research Program, which I actually took over after Debbie Burks had run it for about 9 years. So I came up in that family. And now Julie Ake is running MHRP. So our our center here at the Walter Reed Army Institute of Research is called the Center for Infectious Disease Research. And so our institute took all of the infectious disease programs and wrapped them into one center. So, you know, unfortunately for Julie Ake, she couldn't escape me. She used to be my deputy, and now she runs MHRP, but, you know, I'm her guardian angel one step above. But that's important because many of us in the HIV field, when Ebola became a problem in the West African outbreak, I will tell you, up until that outbreak, there was more concern about Ebola as a potential bioweapon to be used by nefarious people than there was about it becoming a public health concern. Obviously, that was misguided, as Kate and others here know very well. Kate was instrumental in helping to shape the what had come up in the HIV field for community engagement, good participatory practices that UNA and AVAC had nurtured for a very long time. She helped adapt that with Libby Higgs and others to become more of a generalized blueprint for community engagement in pandemic responses. And that was good work that I think we should touch on sometime during this presentation, because there's been some triumphs of science, but I think there's been some far from triumphs in terms of community engagement with the current pandemic. So the Ebola history was I wanted to start there because it began to shift my own understanding of developing vaccines for important infectious pathogens, because even though I was part of the Thai HIV vaccine study, which showed some degree of efficacy in Thai heterosexual populations at moderate risk for HIV infection, that particular approach, when many adaptations were made to the vaccines, so it's difficult to make that bridge, but that thematic approach for an HIV vaccine did not work in Africa in relevant populations. And so I think that's important, because those of us in the HIV field have never really gone like we've done for other diseases. We haven't gone from A all the way to Z. We go from A to about, you know, maybe F, and go back to A again, right? So for a lot of us in the field, in the HIV community, a number of us jumped for Ebola. A larger number of us jumped for the approach to Zika. And we're on the way with some pretty good vaccines, one that we had made at our institute, one that the Vaccine Research Center had made, but the epidemic fizzled out. It's still smoldering in certain parts of the world. But the important thing is that it began to gel the community of people within the government, in industry, in the public sector, in academia, as well as the private sector in terms of pharmaceutical companies. It began to develop the same teams that began to understand how to go from A to Z. So for those of us that are HIV vaccine developers, I learned, you know, by drinking from a fire hose, but how you could actually make a vaccine and take it all the way through deployment and begin to understand what the problems were with deployment. I mean, I would love to be in a position now to have an effective HIV vaccine that was effective, universal, required, you know, minimal cold chain, was one shot, worked for a lifetime against all variants. I would love to be in a position now to have to worry about how to deploy it, because that would mean we would have made it substantial scientific progress. But I can tell you that when it came time for SARS-CoV-2 to be tackled, it was literally the same people around the world that marshaled their efforts to focus on SARS-CoV-2. So that's the first thing I wanted to say. I know a lot of you have grown up like I have in the HIV community, and it was the lessons that we've learned, the attention to community engagement, and the intense focus there at the same time, and not at the expense of the scientific approach, was something that many of us could see from a long way off that we were going to have trouble, because it was obvious to us pretty early on that you know, we were going to be able to make an effective vaccine pretty quickly for SARS-CoV-2. And I remember having some discussions with a number of my activist friends who said, you know, don't forget, once you develop a safe and effective and affordable and deployable vaccine for the world, that you're going to have to figure out how to get people to take it, right? So all of a sudden, all that theory that we developed for for HIV vaccine was put into practice. Why? Because the respiratory virus community was not steeped in good participatory practices. And I sometimes felt like I was a lone voice in the wind telling everyone to read, the second edition of that on the AVAC website, and also to read what Kate and Libby had largely done with the World Health Organization for emerging pathogens, that we were going to end up having trouble. So I can tell you that in terms of what we did with developing vaccines for SARS-CoV-2 or COVID-19, the disease, I think we really did a great job in the testing community. And by that, I mean the studies that were done largely through a new network that was constructed from taking the HIV vaccine trials network, the HIV prevention trials network, and the infectious disease clinical research consortium. That's headed by Kathy Newsel and others that largely dealt with influenza. Putting those three groups together, I think community engagement became a very significant part of that portfolio, and we were able to make a lot of important changes in how the phase 3 trials rolled out. I will tell you that the initial ones supported by the US government were too white, too young, and too low risk. And we had some pretty tough conversations, but because the US government was funding those studies completely, we were able to do the right thing. So we ended up having large community engagements, meetings almost every day, including a centerpiece meeting every week that was headed by Michelle Andrasik, who's a wonderful community engagement expert at the University of Washington in Seattle, and Stefan, and a number of others that really pulled together what we knew about the HIV community engagement paradigm, and then began to work with other sectors. I mean, we're working in meatpacking industries, right? I mean, that's not something we've ever done. And we're working in obviously populations of color, which we knew at that time were at 3 to 4 times higher risk. But, we didn't have as good outreach into Indian country, into populations of color that were also deeply faith-based. So there were a lot of gaps, and I think we closed them relatively quickly. And I'm pretty proud of how the trials ended up being in terms of inclusivity at every level, and the engagements we had with the communities. But that said, right, we ended up with vaccines that were spectacularly safe and effective, but half of the United States alone wouldn't take them. I mean, don't get me started on the rest of the world. I mean, there are problems beyond that. But so what I would tell you is that I think what we did initially, faced with a virus that was largely of a single type, very different from HIV initially. I think we did a great job. Within literally 6 months, we were on the road to making not 1, but 3 vaccines that will be eventually approved at some level in the United States. A 4th was just added yesterday, and more are coming. So, so I think in terms of what we did here in the United States, I think that was a triumph of science. And I think that we were able to build an airplane while we were flying it in terms of community engagement. Where we failed, and just my personal opinion, is I think we had a really poor implementation plan. In terms of logistics, getting vaccines out, cold chains, that was a triumph too. But in terms of reaching important communities at risk and convincing them that this vaccine was important for them to take, for lots of reasons, I think we were really well behind the curve and still haven't caught up. Now, some of that are for reasons that you can imagine. I mean, I haven't seen political divisions in our country since I was a kid living through the Vietnam War era. So we have a lot of mistrust in the system. We have a lot of outlets now that are, spreading vaccine misinformation. I mean, you have individuals that have vaccine hesitancy for lots of reasons. And I just don't think we've come to grips with the crushing need to start putting more money into implementation research. I mean, if you take nothing else from my opening comments, take that one, which is that I think we've proven that unless it's a pathogen like malaria, tuberculosis, or HIV, which are really difficult scientifically to make vaccines for, even though there's progress on all 3 fronts, it was not hard to make vaccines for SARS-CoV-2. And I'm saying that we've had significant loss of people around the world. I had a very significant personal loss in my own family because of this disease. So I'm not minimizing it, but I'm saying that in terms of being able to actually scientifically tackle this dragon, it was not that difficult. And I think, unfortunately, if we were to start all over again, we would still be in the same problems in terms of not really understanding how to effectively reach populations and implement an effective and safe and affordable vaccines for SARS-CoV-2. So I leave that with you as a crying need. And I have continued to beat the drum with funders, especially with the NIH, about how they really need to not look at implementation research as soft science, but a critical science, because one of my colleagues at the University of Maryland said these vaccines aren't 95% effective if only half of the people will take them. They're more like 45%, right? So I think that's a critical point. Let me next just say that, so I was very heavily involved in vaccine development for what we used to be called Operation Warp Speed, and it's changed names a couple of times. Now we call it the H Corps, which is, I guess, boring, but maybe a little more realistic. So we still are continuing to look at now a virus that's beginning to look, I'm not going to say it looks a little bit more like HIV, because as an HIV person, I mean, the amount of diversity we see in SARS-CoV-2 is nothing like you see in HIV. All right. So there are more variants of concern that are coming out, and they are providing a challenge to vaccine developers. That's definitely true. But even that said, even though the vaccines are now less effective, maybe against Omicron in terms of transmission, in terms of hospitalizations and death, these vaccines still work really well. Now, the folks that end up getting really sick tend to be either folks that have problems with their immune system to begin with. Maybe they've been vaccinated and never got a take. This is something we've known for other vaccines in a long time in those kinds of key populations. Or it's in individuals that have had so much vaccine hesitancy they don't want to get vaccinated. So largely, yes, we are seeing hospitalizations and death, but it's nothing like it was 18 months ago. And the vaccines, a recent report suggested worldwide we've saved 20 million lives. That to me sounds probably about right. So I think one of the questions now going forward is, and actually Tracy and I were having this conversation just before we went live. And I was really happy that she brought this up, because I told her I wish that the head of CAAB was in the audience when she said this. She says, well, I've heard that maybe newer variants like Omicron, if you actually made a vaccine with an Omicron, put Omicron in the vaccine itself, that maybe it wouldn't work as well against Omicron, which seems counterintuitive, than using some of the older variants. And I was like, wow, that's just amazing, because there is actually some good evidence that some of the Omicron variants don't really raise great immune responses. They're not really good to evoke immune responses. And, we have data that some of the older variants actually are really good at covering Omicron. And I think the problem is that there are some parts of our field that look at the color of the car, right? So they see a Nissan Sentra that's blue or red. What they don't know is the blue one has got a souped up engine, but they don't want to look under the hood. They just said, well, that's a blue car. The red car must be better, right? And so I think that one incredible challenge that we're going to have coming up in the fall is to try to explain to individuals why they should be taking these newer vaccines, and whether or not these vaccines are going to be, it still takes a lag time to make them. Are we making vaccines right now against variants that we haven't seen, and will they work? So I think a huge challenge is to convince the public that even going back to the ancestral strain, vaccines based on those are still largely protecting us from getting super sick. We may still get infected, but at least now we have drugs, we have monoclonals to treat those people that might need some additional help. But explaining, why a certain kind of vaccine, which has components that maybe aren't intuitively going to work, shouldn't work as well, actually might. That's because the way the vaccine looks and how we characterize it based on Omicron versus Beta versus Delta is less important than understanding whether or not the immune responses, once it's put into your body, those cross-react against a wider range of variants. That's really what you want, right? So at the end of the day, what we're working on at our institute, working very closely with the Vaccine Research Center, a number of government agencies, and a large collaboration with an industry partner, a major pharmaceutical company, we're all working to try to generate universal vaccines, right? So I think what we really want to aim for is a vaccine that would work against not just SARS-CoV-2 and all its variants, but also against the original SARS that we saw about almost 20 years ago, as well as anything that could possibly pop out of a bat. Because if you look at related coronaviruses in bats, there are some that are probably 3 or 4, genetic changes away from being able to jump into us. So you'd like to be able to make a vaccine that ideally predict that whatever we see as a threat, that we would be covered. So we would be able to make a kind of vaccine that would protect us against a wider variety of coronaviruses. So I will tell you that there's a lot of effort in our group and others for this, and we're probably going to start in about a month. We should have the last bit of data that we'll publish in a medical journal looking at our initial first-in-human study. But it looks like it's on the road for that kind of vaccine. We still have a lot more work to do, and that vaccine is not going to be the one that is going to be pan-protective, but it's beginning to tilt us in that direction. So that's what I want to leave you with, sort of like 3 major points. One is community engagement and understanding how to implement vaccine campaigns is underappreciated, underfunded, and it will continue to stymie our ability to do global public health until funders understand that they have to put a lot more money into advocacy and a lot more money into implementation research. So, and I'm telling you that from the standpoint of being an MD, PhD, hardcore scientist. I watched it happen. I just literally saw it coming, and we watched our ability to implement this vaccine, even in our own country, where resources were not as much of a problem. It didn't work well, right? And it didn't work well because we didn't pay attention to implementation research. The second point is that we can make vaccines against these coronaviruses, but being able to sort of stay ahead of them has become difficult. And so we're going to need to be able to put ourselves into a position to try to think ahead of where Mother Nature is headed, right? And the third point is that those of us in the HIV community can feel proud that we have, from time to time, we have put our arms around other parts of the field. And a lot of things that we've learned in terms of immunology and science, community engagement, are now being increasingly proselytized across other fields. And I think that really, it strengthened our field, because I've now learned a lot more concepts outside of HIV that were helpful for me for HIV and vice versa. So I think our community, our tent's gotten wider. So those are the 3 things I want to leave you with, and maybe I'll stop there.
Thank you so much. Everyone, please feel free to put questions in the chat. And while you're taking a minute to do that, I'm going to take the moderator's prerogative and ask you a couple of questions.
Yes, ma'am.
The first one is, if we're looking forward to vaccines that would be effective against future variants and even other coronaviruses, are we using a different approach or different targets? What's different about the vaccine you and your colleagues are working on than the ones we have now?
Yeah, so the answer is yes on both points. So in terms of different targets, I'll start there because I think that's probably an easier one. Almost exclusively, and certainly exclusively for those vaccines that are actually being used as public health tools. All vaccines being used as public health tools are focused on the part of the virus that actually grabs onto one of our, into one of our target cells in us and gets itself in, right? So in HIV, we would call that the envelope protein. In SARS-CoV-2, it's called the spike protein. So that's the initial part of the virus that sticks out and grabs onto our cells and gets in. It gets in through a receptor that's called ACE2. And importantly, that seems to actually work pretty well. But there are groups now that are taking the approach saying, well, maybe we should look at the ability to put in other targets other than spike. And the one that most often comes up is something called nucleocapsid. Nucleocapsid is a part of the virus that actually, it's a protein that sticks onto its genetic material. And my guess is that would probably work better to develop T-cell immunity, which may be important in having vaccines that are more durable. I think the problem with the current vaccines is that even under the best case scenario, they tend to wear off in about 5 to 6 months for the most part. And having to boost every 5 or 6 months that's not an effective public health strategy. So part of our approach is to ask whether or not we should be using spike plus a different approach. So there are a number of groups now that are just beginning to do this, to look at other targets other than spike. So that answers Tracy's first question. In terms of approaches, yes. And so the traditional, I think you just saw it happen. mRNA vaccines have been in the works for well over 15 years. And it just so happened the science is just at the right point when SARS-CoV-2 came along that we're able to work very quickly. So mRNA vaccines are well ahead of the pack. It's true that Johnson & Johnson developed a viral vector, an Ad26 based approach. That's just taking a virus that usually causes a cold, and they used it as a vehicle to give a vaccine. And even though that actually worked well, there were problems with that, concerns with that vaccine because of side effects, right? And so I will tell you that I think that vaccine is safe, and it saved a lot of lives, especially in countries like South Africa. In the United States, it just, rare problems, but definitely significant problems with clots were noted. You're talking about maybe 10 cases out of 100,000 people. So when the pandemic is raging and people are dying, 5,000 a day, which was about the number in the winter of 2020 to 21 range, that's a different public health decision than it is today, when the number of daily deaths in the United States are more like 300. So I think that's why having choice being broad is important. And lastly, the Novavax vaccine that just got approved, that's based on protein technology, which has been around for a very long time and has a lot of advantages. It's in terms of not developing the allergic responses that have been seen with some of the mRNA vaccines. So there are approaches like that: viral vectors, proteins, mRNA, all those kinds of approaches are now being explored. If you look at the Novavax, it actually has a little bit of a twist on a protein vaccine, because it actually assembles itself into what's called a nanoparticle. And so instead of being sort of an isolated protein, a number of the proteins collaborate and they get together and make sort of like a beach ball, right? And there's some advantages theoretically to that kind of platform, but that's brand new science. And so I'm excited about that. And I also am excited about the use of new adjuvants. Novavax is using a modern adjuvant, not like the ones that were used 50 or 60 years ago. They're using some newer ones. So is Sanofi Pasteur, which their trials are just being unblinded. It looks promising. So I think you're going to be looking at a whole sort of new approaches, Tracy, which is you're going to be looking at traditional approaches like protein vaccines, maybe with tweaks to make them into nanoparticles. You're going to looking increasingly at other kinds of viral vector approaches. The viral vector approaches seem to be good at making the cellular part of the immune system durably activated, and that may be a good thing. So we still have a lot to learn, but I will tell you that looking at something other than spike, yes, target is being investigated, as maybe that's not the way the story should end, and the way that we actually give vaccines in terms of how they're delivered, what we might deliver them with, all that is being explored scientifically. And I'd say that to me is, and I keep on coming back to this because if I try to tell someone in the community, especially one that maybe has had historical mistrust or has been abused by the traditional medical systems in either our country or overseas, telling somebody, well, we don't have a lot of experience with this, but we're really excited about it. That may sound good to some people. It may sound like, oh, great, you want to use me as a guinea pig, right? So what I will say is this, is that one of my real heroes and a new friend in this was Dave Wilson, who heads the Tribal Health Research Office at the NIH. And what he taught me is that in Indian country, there was a lot of concern about, Western medicine, if you will, and the predation of American Indian, Native Alaskan, and Native Hawaiian populations. And we also knew at the same time that that those same key populations were at 5-fold risk of having bad things happen if they got SARS-CoV-2. And so at the end of the day, that particular population, because of a number of factors in terms of the tight-knit association between tribal associations, between Dave's stewardship, the Tribal Health Research Office, which reports directly to the NIH director. At the end of the day, the vaccine uptake in Indian country, which is the proper term to describe that collection of key populations, their uptake was much higher than it was in the United States in general, because they were able to effectively use providers, trusted advocates, tribal elders. I mean, they really did it right, and that story has not been told. I mean, I know it because I'm, Dave and I are buddies, but I think that's going to be really important going forward is understanding that certain parts of our communities at great risk were able to do the right thing because it was explained to them that, yes, you may be taking risk going to an experimental vaccine study, but our communities are at much higher risk. So in terms of, altruism and other, part of the inducements for someone to join a clinical trial, there was that sense that this is right for me to do because the government isn't going to test this vaccine on us so that they can use it for everybody else strictly. They're going to test it on us because we are a key population, an intended use population, and it makes sense for it for us to be initial volunteers, because if they test it in other communities, it may or may not work with us. We don't know that, right? So that was part of the dialogue for why it was so important to have diversity in trials. And it wasn't just a nice thing to do. It was because the intended use of this vaccine had greater impact in communities, in Hispanic communities, in Indian country, in Black communities. That's where the risk of disease and the risk of death was much higher. So it was actually something the FDA finally put into draft guidelines, saying we can't just look at populations that are easy to test, young, white, affluent. I mean, that may or may not convey to how the vaccine works or how safe it is in other populations that need it even more. So I think this is something going forward that the activist community really needs to get behind, because this war is far from over. And there are a lot of lessons we've learned in SARS-CoV-2 that I think are going to continue to be important for every campaign that we have against vaccines. You're muted.
Thank you so much for that. And, to touch on this, particularly engaging diverse populations in research, trials are going to start looking really different now, because we can't really compare a vaccine to a placebo. It's not ethical, since vaccination is a standard of care. So one thing I think that was problematic about the original trials was participants weren't tested regularly while they were being followed unless they had COVID symptoms, right?
Yes.
And trials define those symptoms differently. And up to 40% of people don't even have symptoms. So it could have made the vaccines look more effective at preventing infections than they actually were. Going forward, what are your thoughts about trials that, besides doing the work of really thoughtfully engaging with different communities and their leadership and their stakeholders, how are trials going to be look different, and what can they offer to people who participate, besides just some money?
Yeah. No, those are all great questions. So let me try to tackle them in order. You're absolutely right. And a decision was made by those trials that the U.S. government funded. Now, I remind you that Pfizer funded its own initial studies. The government bought their vaccine, but Pfizer decided to use its own money and went its own way. They crossed the finish line first. Maybe some dark humor for those of us in the government, not being surprised that the private sector works faster than the public sector, but it was only by a few weeks. But still,
although keep in mind, BioNTech got a huge grant from the German government, Pfizer's partner. So
they did. So but I think at the end of the day, you know, the U.S. government made a decision, and they published in the spring of 2020 what they're called the tenets for the large phase 3 trial. So there were certain guidelines for how the trials needed to be constructed, right? And obviously there was some wiggle room, and you've addressed that. But their decision was made early on, and it was not everyone agreed, but a decision was made early on that they should only look at symptomatic COVID infection. Part of that was because you had financial constraints. Remember, the government took a Noah's Ark approach, right? 2 by 2, 2 protein vaccines, Sanofi, Novavax, 2 viral vaccines, based on adenoviruses. So that was AstraZeneca and Janssen, part of J&J, and 2 mRNA vaccines, Pfizer and Moderna. So, I mean, the government was putting well over $10 billion into that science. And so some accommodations had to be made to be able to do the trials efficiently at reasonable cost. And because literally, at the time, the death rates were climbing well into the, into the 3,000 to 4,000 range, there was an intent to really get the answers fast. Okay, and I think in that sense, we were successful. If you were testing literally everybody, regardless of symptoms or not, you would have learned some critical things about transmission. There's no doubt about that. But imagine doing that. I mean, so Moderna was done completely within the United States, and Janssen was done much more internationally. Novavax somewhere in the middle, and Sanofi Pasteur largely outside the United States. You haven't seen a lot of those data yet. They're coming, but their vaccine looks good. Tested against Omicron. I mean, literally tested in an Omicron wave. Their vaccine looks really good, and the vast majority of those data came from outside the United States, and they came in places like, I actually visited one of their sites in Ghana, which is right on the border with Burkina Faso, just about 6 weeks ago. I mean, it is austere up there, and so just being able to get folks from where they lived in their villages into the Navrongo vaccine research facility, not trivial. And so, and doing that with the temperatures average, about 100 degrees Fahrenheit. So there are logistical problems for testing people more frequently. Now, that said, after the trials were initially unblinded with Moderna and Pfizer, there was a huge effort with the HVTN, HPTN, the NIAID to try to do transmission studies. And this is where I was learning hand over fist from people that were like Kathy Newsel, who's become a new hero of mine at University of Maryland, because she was saying, there were some people saying, let's do this on college campuses, right? And so we'll swab these students every single day, and we'll get to learn, and we'll do it in the context of a vaccine campaign. And Kathy's like, well, we tried that before in flu, it doesn't work too well, No, I mean, I was a college student once, and I got a 24 and 27-year-old at home. So my experiences are recent. I'm like, you think they're really going to do that every day? But there was a huge debate about that. Kathy's saying, do household studies. Others are saying, no, don't do it that way. I will tell you that we weren't so good at trying to ask some of those questions early on by doing transmission studies in the context of as we rolled the vaccine out into those communities. So I will tell you that even though a huge amount of smart people were involved in that, that was not a success story. You haven't heard a lot about it because, well, they say, success is a lot of suitors and failure, an orphan. So it's hard. It's really difficult. And I look to the influenza community because people like Kathy grew up in that tribe. And they are really good at figuring out how you could ask those kinds of questions. I do think, Tracy, that it's really important. I think that as people struggle with long COVID, what drives people nuts is when they say, well, look, hospitalizations and deaths are way down. What are you worried about? Well, talk to people with long COVID who still have brain fog 6 months in, those are the real issues. Now, I would tell you that you could say, well, there's Paxlovid and there's monoclonal antibodies. Well, ask what the uptake of those reagents are and how difficult it is to get an infusion if it's a monoclonal. So Paxlovid has got its own problems. Maybe the treatment course is too short. So I think your question is incredibly relevant still, because long COVID is, it's real, and it's something that we have to struggle with. I'm 65. If I get COVID, I am literally driving to CVS while I'm calling my doctor saying, call the script in for Paxlovid. I'm taking that in a cold second. Why? Because my immune system is tired, Now, my 27-year-old, he got infected, and 2 days later, he's good to go. His immune system's a little younger. So, I think we have to be concerned about the issue of transmission, because we just simply don't have the resources to get some of these new experimental treatments out. And we still don't really understand how effective they'll be in blocking long COVID. So that's why I think you're pushing hard on the fact that we need to look at transmission still is an important point, because there are long-term sequelae of these diseases. And it really shouldn't surprise you that there are. There are long-term sequelae to Ebola infection. There are long-term sequelae to any of these approaches. We've known this historically from diseases like shingles, right? So, I think that's a really good point, which is why I keep on saying the war is not over, right? Even though I think we're all exhausted with this pandemic.
Thank you so much. And I just found a question in the Q&A from Matthew, which is, as we think about this next phase of vaccines, are we also looking at the ability to make these vaccines in regional places around the world? How do we avoid some of the production bottlenecks caused by the first vaccines?
Yeah, no, that's a good point. So I'll give you some anecdotes. Again, the HIV person in me says, we have been dealing this with every phase 3 trial. There's always a tech transfer agreement at the end saying, if this vaccine works, we're going to provide it first to the people of South Africa. If that trial was done there, and then we'll roll it out across the continent. Well, unfortunately, we haven't gotten to the public health implementation phase for that. But so the anecdotes are about a week and a half ago, BioNTech signed an agreement with the government of Rwanda, and they broke ground on a vaccine production facility in that country, in Kigali. So there's going to be of course, it's probably will take them 18 months at least to get it built. And my guess is probably 3 years before it could be fully operational. But BioNTech has made a commitment to make a vaccine production for mRNA on the continent of Africa, right? So that's one anecdote. And the other anecdote is the International Vaccine Institute, headed by my colleague Jerome Kim in Seoul, Korea, just announced that a protein vaccine, which is also a nanoparticle, it's one very similar to the one that we're making in our institute. It blasted through phase 2 and 3 trials in Korea and elsewhere, but especially in Korea. And SK Biosciences, which is a Korean company, is going to make it. And my guess is that will probably be a vaccine that will be much more likely to be used in the Asian market. So you're already, those are two anecdotes that's not based on me doing a careful landscape, but I'm just telling you from what I'm hearing. That is no longer just rhetoric, right? So BioNTech is spending real money to do it in Kigali. SK Biosciences is probably going to be muscular in most of Asia. I mean, China is always going to go, it'll go it by its own approach. I could tell you that the government of Thailand is intensely interested in making some of the Chinese vaccines. It wouldn't surprise me that they start making the nanoparticle vaccine that IVI has brought forward as a public health tool, because there's such strong collaborations between Korea and Thailand. I mean, Jerome's critical to that. Jerome was really the architect of the success of the Thai trial for HIV, and spent a long time in Thailand, and now he's the Director General of the International Vaccine Institute in Seoul, Korea. So, he's very well known in both those countries. So I think you're beginning to see that. That goes back to my earlier point about the fact that we in the HIV field crossed over, vice versa. I think both sides are being helped out, but I'm happy to see already there's at least two good examples of tech transfer not being to the rest of the world, not being just empty rhetoric.
No, thank you for that. I mean, where there are those good examples, I think there are also some that are really problematic, like the license that J&J gave to Aspen Pharma in South Africa. And it was fill and finish, not complete manufacture. But even though South Africa specifically had purchased vaccines, they took the ones that were fill and finished in Africa and delivered on EU orders first, even though EU was buying them at a lower price than South Africa. There was a huge New York Times, pretty scandalous story. And I think some of the movement to waive intellectual property, it's really, really important. But since we're talking about global access, I noticed that you didn't mention the inactivated virus vaccines. And I'm wondering, there was this idea that really circulated, mRNA is so much better, but now that we've had some time, could they not be generating immunity that's just as good as the mRNA vaccines? And is that not also a promising approach?
Yeah, well, I mean, I didn't talk about them a lot, only because, from my perch in the U.S. government, where I was heavily involved in 6 campaigns to develop vaccines, inactivated vaccine was not one of the Noah's Ark approach. So those animals weren't taken on the ark.
Didn't get on the ark.
But of course, but of course others did take that approach, especially China, right? So that getting access to all that data has been fraught. So, I don't have the same level of intuition scientifically about those products as I do with the others, only because I just don't have the same access to all of the data, right? But what I will tell you is that have inactivated virus vaccines worked well against other viruses? Yes. I remember I told you that we were involved in making a vaccine against Zika, which actually looked pretty good. And that was it. We grew up Zika and Vera cells. We inactivated it with formalin, formaldehyde, purified it, and it worked pretty well in terms of initial safety and how it looked in terms of immune responses. We never got to the point to test it in an efficacy study, and we were headed that way. But the epidemic quickly waned, and so both we and the VRC, who are using a DNA vaccine approach, never got to ask that question. But my point is that if you were the kind of person who studied flaviviruses like dengue and Zika and other kinds of viruses of that type, you would say, yeah, of course, inactivated viral vaccines make a lot of sense. So just to take the argument further, I've talked about choice being so important here, right? Look at polio, right? So the initial polio vaccine made by Jonas Salk was an inactivated virus vaccine, right? Worked great. Of course, there was a very notable incident where the inactivation wasn't good and people got hurt. There were problems. So then Sabin took years later, Albert Sabin developed, a live attenuated approach. And that's the one I took as a first grader and in a sugar cube, at Pleasant View Elementary School in Kensington, Maryland, when I was a kid. But, so increasingly, because of the problems with live attenuated, you get breakthroughs, maybe 1 in a million. Well, if the only polio cases are coming out of Burkina Faso, Pakistan, and Nigeria, because people aren't vaccinated well enough, or if they're coming from those breakthrough cases, maybe it's time for us to switch back to the inactivated vaccine, which is happening, right? So my point is that, your question that you raise, I think is going to be one of safety versus efficacy, logistics. So I do think that there is probably a role for vaccines of that type, because as long as their manufacturing is well controlled, inactivated viral vaccines have in a number of different cases across a lot of very different virus types have been shown to be safe, inexpensive, really inexpensive, right? And the storage is simple. So if you're talking about a vaccine campaign for austere parts of the globe where you don't have a lot of refrigerators, and resources, maybe $10 a year per capita for the entire public health of a country, then those kinds of vaccines might make sense, which again, I think at the end of the day, it's not just arrogant, it's scientifically implausible to say that any one vaccine is going to be best for the entirety of the planet. So I 100% agree with you that I think inactivated vaccines have their place. And the fact that the US government decided not to fund them, I don't think is particularly relevant. It doesn't mean that that anyone anticipated they wouldn't work. And there are, a significant amount of experience now with those vaccines in other places in the world. So I think that as the situation evolves, I think that you saw that the viral vaccines took a big hit, and maybe some of that's unfair. But trying to convince any given person in the United States to take the Johnson & Johnson vaccine, when the CDC puts out an announcement saying, take it only if you have no other choice, that's a tough one, right? So that's why I think there's still going to be markets for the protein vaccines like Novavax and soon Sanofi. And I think the inactivated vaccines will definitely have their role. Whether or not that role will be soon or ever in the United States is hard to say, but it's already being used extensively in Asia. And I think we just need to continue to keep an open mind.
Well, I can't thank you enough. And I see a comment in the chat about some real interest in the HIV vaccines field, current reliance on Moderna for mRNA technology, what needs to change and when. And I realize we're at time right now, but I have a feeling that we're going to be reaching out to you and drawing on your expertise again for this and possibly other updates. And I cannot thank you enough.
All right. Well, I know Stacy pretty well. I'll write to her so she doesn't get mad at me. But I'll do it on email.
Thank you so much for joining our CAAB. Really appreciate it.
All right. Happy to be here. Thank you.
Thank you.
Bye-bye, guys.
Bye-bye.